Hello, my name is Meha Sridhar, and today I'll be discussing research regarding abscisic acid and its effects on glucose homeostasis. Glucose is an essential carbohydrate that we all have in our bodies, and keeping a healthy amount of glucose flowing around our blood is essential for keeping us all healthy. However, there are often anomalies with the current process that we have within controlling our glucose levels. This can lead to too little glucose, hypoglycemia, or too much glucose, hyperglycemia. Insulin is the only hormone in our body that we know of so far that is used to mediate the amount of glucose that we have. Unfortunately, high glucose and insulin resistance have become big problems in our world today. People regularly suffer from hyperglycemia and often have difficulties treating it due to insulin resistance. Today, I hope to present a potential way to help solve this problem. Abscisic acid, or ABA, is a conserved plant hormone that mainly specializes in stress responses. It plays a major role in deciding when seeds should germinate and to ensure their survival, and making decisions to ensure the plant's survival during drought. Since ABA is produced in plants, it's common in many of the fruits and vegetables that we commonly eat as well. It has a long evolutionary history dating back to the time of single-cell organisms. This means that ABA is present and produced in many plants and animals, including humans. If it is present and produced in humans, then it raises the question, ABA must play the role in humans. And what is this role? While ABA's role in plants is much more well studied, recent studies are showing that ABA is able to regulate glucose levels without insulin. Most cells take in glucose by using insulin as a key to open the glucose channel to let in glucose. So far, this is the only known way to manage glucose levels in our body. However, there are new studies showing that ABA is able to initiate a different signaling pathway to allow cells to take in glucose. The first study was done on rats, where groups of rats were given a glucose fluid and either synthetic or naturally extracted ABA or no rate ABA at all. The rats that were given the glucose fluid displayed a lower glycemia profile, and they also had unchanged levels of insulin, showing that ABA was able to play a role in glucose homeostasis without affecting insulin in our body. There are two types of adipose tissue or fat. White fat cells are made for storing fats, and brown adipocyte cells are made to produce heat. Acquired insulin resistance can lead to obesity and eventually develop diabetes because insulin is not able to play a role to help transport the glucose into cells. This leads to an increase of white adipocytes and can cause many problems in people. Another study done on rats showed that the rats had less glucose in their blood and higher glucose tolerance. The rats also had less white adipocytes growing and increased expression of the gene that stimulates glucose uptake. This shows that the ABA is helping with controlling the levels of fat in our bodies and is helping the rats have a more healthier glucose processing system. Due to the correlation between type 2 diabetes and obesity, targeting obesity can help lower the risk for GTD or help lower its side effects. ABA can also work around insulin resistance, providing an alternate route to battle high glucose levels. This proves that ABA could possibly be the key to solving various problems around glucose homeostasis. Thank you for listening.